Alright, chapter 6 is all about the circulatory system. And you can look at the objectives and whatnot on your own. However, we're going to start on the overview page. So basically the purpose of the circulatory system helps carry oxygen and basically the food of the body to all the cells, and also picks up all the waste and the carbon dioxide and brings it away from the cells and delivers it to whatever waste area it needs to go to. The circulatory system is responsible for the coagulation process. It's also responsible for helping defend against disease, helping regulate body temperature, and it's made up of two parts, the cardiovascular system and the lymphatic system. That's usually the one that most people are unaware of, that is part of the circulatory system. So the heart is, of course, the major structure of the system. It's made up of 4 chambers. You have 2 atria and 2 ventricles. It's surrounded by a pericardium, and there's basically a right side and a left side. So when we talk about the heart, we're usually going to be talking about which side, and then whether we're talking about the atrium or the ventricle. All right, so we'll talk about the layers of the heart. You have the epicardium, the myocardium, and the endocardium. I think that this is supposed to play a little video. Let me see if I can make that happen. Let us see. I go. It does not. Bummer. Oh, there it is. As you can see, this little video shows the blood coming in. Oh, we'll have to wait for it to start over here. Alright. So, it comes in from the body, right? Lands there. Goes out to the lungs. Comes back. Alright, and gets pumped out to the body. Pretty cool. The upper chambers here are the atria. The lower chambers are the ventricles. So we call them the receiving chambers and the pumping chambers, okay? Your book goes over all the valves. I'm not going to test you on that, but it is important to know them. Okay, so the chambers of the heart, you have this table in your book, tells you a little bit about them. We'll go over that, the order of the blood flow and all that in a little bit. All right, so moving on. Coronary circulation. The heart doesn't actually get its blood or its oxygen from the blood that it pumps through it. It gets its blood from the right and left coronary arteries. So there's arteries that wrap around the heart, and that's actually what supplies the muscle of the heart itself with its oxygen and nutrients and all that good stuff. So when something happens to these coronary arteries, this is when we hear someone having a heart attack, which is technically called a myocardial infarction. It's because something has happened to these coronary arteries. Either they've been obstructed, you see this term here, ischemia, or they have basically, been obstructed for so long or have such a complete obstruction that the tissue actually dies there due to a loss of oxygen. So this is showing that again. Isn't that pretty cool? Yes, it is. All right, so the cardiac cycle, you do want to know that, the difference between systole and diastole, what the electrical conduction system is. So you have right over here, you have this picture. Where is it at? You have the sinoatrial node. Oh my gosh, there it is. Right on the top here. It's on the atrium, right? The right atrium, which is where the blood comes in. This is what initiates the heartbeat, the contraction of the heart, right? So this is the pacemaker, the natural pacemaker. So if you've ever known somebody that had a heart attack and then after had to have a pacemaker put in, it's because when their coronary arteries got all messed up, this part of the heart is what died, and so their natural pacemaker, their natural piece of equipment basically that synchronizes the whole contraction of the heart died, so they have to have a fake one put in, a pacemaker put in. So that's actually what they're replacing when they do that. You guys can look at this. This is EKG stuff. And it talks about the actual lub-dub sound of the heartbeat, how it's created by the valve. So the atrioventricular valve closing is the first heartbeat sound that you hear, and the semilunar valves closing is the dub sound, the second part of the heartbeat sound. Kind of cool. You can learn these little facts here, but I'm not going to test you on these, so I'm going to go past that. You do want to know what a pulse is, right? And that's part of the reason we don't feel with our thumbs, because we have a little pulse in our thumbs. You want to know what blood pressure is? It's the force exerted by the blood being pumped by the heart on the walls of the blood vessels. So you guys have all had your blood pressure taken. All right, so now we're going to talk about specifically the vascular system, which is actually all the blood vessels through which the blood is circulated. And there's two divisions. You have pulmonary circulation and you have systemic circulation. So pulmonary circulation is where the blood leaves from the heart, specifically the right ventricle, and goes to the lungs, where it drops off its carbon dioxide and picks up its oxygen, and then it comes back into the heart via the left atrium. Because remember, the atrium is always receiving blood, whether it's from the body or from the lungs. And the systemic circulation, that's going through the whole body, the body system, if you think of it that way. The rest of the body is the systemic circulation. And so this is a very simplified picture of that. So you have the blood coming in from the body, from, say, the head, and it goes to the right atrium, and then it travels out of there towards the pulmonary, doing pulmonary circulation here, picks up oxygen at the lungs, comes back in through the left atrium, goes down to the left ventricle, is pumped out through the whole body, whee, yay, right? And then it comes back to the body via the right atrium and does the whole thing again. Okay, so you have a boatload of blood vessels. According to this, you have 250,000 miles of blood vessels, most of which are capillaries. That's a bunch of capillaries, don't you think? So, arteries are the blood vessels that carry blood away from the heart. I always remembered A for arteries, A for away. So arteries carry blood away from the heart. And in most textbooks, you'll see them shown in red. Okay, it doesn't mean that arterial blood is red and venous blood is blue. It's just the accepted way that people have decided to do that, to show that, to represent it, is that usually the more oxygenated blood is, the more red it is because of the hemoglobin and the iron and all this stuff, the way that the chemical makeup of our blood is. But just so you know, as we go back a slide and you see this blood going away from the heart to the lungs, those are pulmonary arteries, and they are deoxygenated blood. So this is going to be generally the only time you see them, any arteries depicted in blue, and some veins right here. This is the pulmonary vein depicted in red because it's very well oxygenated. But remember, veins go to the heart, arteries go away from the heart. So don't let that confuse you. So arteries have very thick walls compared to veins, and that's because they're under pressure from the contraction of the heart and the ventricles. Generally, we can say that arterial blood is, let me go back, bright cherry red in color. Not always. Sometimes you will find, you can do an arterial stick and you'll have blood that's really, really dark in color. And the reason for that has to do with the cardiac output and the patient's respiration rates and a whole bunch of other things. So there are situations where the color is not going to be a good indicator of whether or not blood is arterial. We always want to look for, if we're wondering if we've collected arterial blood, we want to look for the blood pulsing or spurting into our collection tube or into our syringe. And we'll talk more about that too in chapter 12. I think it's chapter 12, the arterial chapter. So big arteries break down into smaller branches called arterioles, which then break into smaller branches, which are called capillaries, as you guys know. And then that'll eventually feed back into veins. The largest arteries, the aorta. I'm not going to test you on that, but it's fun to know. The other types of vessels we have are veins. And so veins return to the heart, okay? They carry blood that is usually low in oxygen, except for the pulmonary vein, as you can see there. Usually the blood is darker in color. It says it almost has a blue or purple tint, but you can have very red venous blood, as you guys have seen in lab, but it is generally a little bit more darker in color than arterial blood. The walls of these vessels are much thinner, which means that they can collapse. That's something we've talked about a little bit in class already, what that is. And the blood flows through the veins, not via the contraction of the heart, but actually through the movement of the skeleton. And then the placement of a bunch of different valves that prevent the blood from flowing backwards. And then, as you can see, through pressure changes in our abdominal and thoracic cavities that happen while we breathe. So basically just breathing and moving helps our blood flow back through our veins to our heart. And those, just like the arteries, break into smaller branches called venules, and then those feed into capillaries. The largest vein is the vena cava, whereas the longest vein is the great saphenous vein in the leg. It's down here somewhere. All right, so capillaries, we've talked about these a couple times, especially in chapter 10. These are 1 cell thick vessels. They connect the arterioles and the venules. This is where the blood kind of mixes together and closely resembles arterial blood. Remember, we talked about this already. This is where all the exchanges of gases and nutrients and waste occur. Our blood vessels have 3 layers. So we have 3 different layers of the walls of the arteries of the veins. The internal space is called the lumen. And then we've talked about valves. And this is a picture showing, I think this is an artery on the left and a vein on the right, and you can see the difference in size of the smooth muscle that lines each one of these and the connective tissue around it. It's kind of cool. This would be a transverse slice of an artery or vein. This is just another representation showing how it goes from a big artery, and then that's right here, and that breaks down into an arteriole, which then breaks down. As you can see, the little box shows a capillary right here. So it's pretty cool. This picture of the vein shows the valve right there. So how the blood doesn't, so once it flows this way, it doesn't flow back. And this is a picture of how the muscles and the skeleton would help move your blood through that. So on page 156, you can see a box 6.2 that shows the flow of blood. You do need to know this. I should be able to start anywhere. If I started at left ventricle, you should be able to pick that up, that from there the blood then goes to the aorta, to the arteries, and so forth and so on. Most of the time you're going to start with the right atrium, and we're going to say the blood arrives from the body in the right atrium of the heart. It is then goes to the right ventricle, where it is pumped into the pulmonary artery to go to the lungs, where it picks up oxygen, and then it comes back to the heart via the left atrium, and it goes into the left ventricle, where then it is pumped out through the aorta to all the arteries, which break down to arterioles, which go to capillaries, where all the gas exchange occurs. And then it starts its journey back via the venules to bigger veins to the superior of inferior vena cava to the heart, where it arrives back in the right atrium, and then it repeats. So you should be able to know this. I should be able to list any 3 of these things. If I said put these 3 things in the order of the flow of blood, and I said capillaries, venules, and arterioles, you should be able to tell me which order that those go in. So for example, that one would be arterioles, capillaries, venules. So you do need to know this. And I do have a video. I don't know if it's going to play here. Let's see.
The heart is a pump which must circulate blood through 2 different but interconnected vascular systems.
When you watch these
the smaller
PowerPoints,
of these systems is the pulmonary system.
Watch this on your own.
Blood returning from the upper part of the body is delivered to the right atrium of the heart by the superior vena cava, one of the body's 2 largest veins, while blood returning from the lower part of the body is delivered to the right atrium by the other major vein, the inferior vena cava. Contraction of the right atrium in each cardiac cycle forces blood into the right ventricle. This is followed by contraction of the right ventricle, which pumps blood into the pulmonary artery, sending it on through the blood vessels of the lungs. As the right ventricle contracts and pressure within the right ventricle rises, the tricuspid valve situated at the opening between the right atrium and right ventricle shuts, preventing any backflow. The pressure generated by contraction of the right ventricle soon opens the pulmonary valve and blood enters the circulation of the lungs. After passing through the circulation of the lungs, the blood, having been recharged with oxygen and having rid itself of carbon dioxide, is returned through the pulmonary veins to the left atrium. The left atrium, too, contracts, forwarding blood into the left ventricle in order to fill it before it contracts. As the powerful left ventricle contracts, the mitral valve shuts, preventing backflow into the left atrium. The aortic valve opens and blood is forced into the aorta, which distributes it to the rest of the body apart from the lungs. As the contraction comes to an end and pressure in the aorta falls, the aortic valve snaps shut to prevent backflow into the left ventricle.
Alright, so that's a pretty cool video, and you guys can access that video from your textbook's website as well. Alright, so moving on from the heart and the vessels is the other vascular anatomy that I have decided, and your book has decided, is phlebotomy related. So the main one that you guys should all be very familiar with is the antecubital fossa, or the AC. right? This is our first choice for venipuncture. This is where our major veins that we really like are. They're easy to locate, they're easy to stick, they're naturally well anchored, it's safe, all those kinds of things. So you should already be pretty familiar with this part because we've been doing this in lab all the time already. So our median cubital vein is our first choice of the three. It's in the center, it's large, it's well anchored, it's easy to puncture, and least likely to bruise. So the cephalic vein is going to be in the lateral aspect of the AC area. It's our second choice. It's a little harder to palpate sometimes, but it's pretty well anchored in place. On your obese patients, this is a good one to look for. It's usually easier to palpate. On your basilic vein is your third choice. It's going to be on the inside one over here. It's easy to palpate, but it's usually not very well anchored. It's closer to nerves, and it's closer to the brachial artery there. So it's not our first choice. It's our third choice, but it is still on the top three before we would go to the back of the hands or somewhere else. We can look on the back of the hands, the sides of the wrist, but never the underside of the wrist. And, that's because we have a lot of other structures and things there, and if we were to cause a hematoma or something to form there, we could cause pressure injury or compression injury there against tendons or nerves or something like that. We don't do legs, ankles, and feet unless a doctor orders it, and then depending on where you work, you may or may not be responsible for actually collecting those. But the risk of phlebitis or thrombosis, circulatory issues, problems with healing, things like that, they're all different reasons why we do not generally draw from the lower half of the body. Arteries are not used for routine collection. It is more painful, it's more risky. Usually they're only used for arterial blood gases and are collected by respiratory therapists. So, what is the blood? What is it made up of? The blood is made up of fluids and cells. It's thicker than water. It's salty to taste. It's a little alkaline. And we each have about 5 liters. Remember, that's like 2 liters of soda, right? It's about that much. So inside the living body, we call that in vivo. The fluid portion of blood is called plasma. And the cellular portion we call the formed elements. And that's our red blood cells, white blood cells, and that kind of thing. So when we look at the makeup of that, as you can see here, fluid is about 55% and the cellular portion is about 45%. We can say that a little over 1/2 of the blood specimen is going to be the serum or plasma once we spin it down, and the other half is going to be the formed elements. So just so you can keep that in mind, if you're told by the lab tech that you need 2 milliliters of plasma, you're going to need about twice as much of that in blood, right, before it's spun down. So it's just something to keep in mind. Just a little picture to kind of showing all the different things that are in our blood. So plasma is 90% water, 10% other things. And those other things are gases, minerals, proteins, nutrients, waste, and hormones, vitamins, drugs, and other chemicals. Okay? The formed elements are made up of red blood cells, which are called erythrocytes. They are the most numerous formed element. Carry oxygen from your lungs and everywhere else. You have hemoglobin, it's the main component of that. Iron contains this pigment that allows the transport of gases, so it's pretty cool, right? It's produced in bone marrow. They have an intravascular function, meaning they don't leave the blood vessel system. They stay they get into the blood vessels and they stay there forever until they die 120 days later. Okay? Then you have leukocytes, which are white blood cells. These do have nuclei, so when you look at them under a microscope, you can see a nucleus on these, whereas red blood cells, you don't see that. They're also formed in the bone marrow, and they obviously, as we all know, help fight pathogens in our body. And they have an extravascular function, which basically means they leave the blood vessels, they leave the capillary walls and go hang out in tissues and stuff like that. So they really don't spend very much time. They take the highway. The blood vessel is a highway. They get to where they're going, and then they go elsewhere. They only live about 13 to 20 days. Your body's regularly making new white blood cells. There's different groups of white blood cells. You guys can look at these on your own. There's all these different kinds of pictures, all the different kinds of white blood cells, and they have specialized functions and features that go along with them. So we also have thrombocytes, which are platelets. They are the smallest of the formed elements. They come off of what's a larger cell called a megakaryocyte, which is found in the bone marrow. And these are essential to coagulation, and they only live about 10 days as well. And they also, like the red blood cell, have an intravascular function. So your body's regularly making these as well. All right, so moving on, we're going to talk about blood type. Now, if you haven't noticed already, in the YouTube supplemental playlist, I have at least 2 other videos on determining blood type and understanding how this works. You guys are welcome to watch those. We just need to understand the very simple basics of this, though. So what you need to know here is that the blood type is determined by the presence or absence of antigens on the surface of the red blood cell. You need to know the definition, okay? It's important that we get the blood type correct. If someone receives a transfusion and the blood types don't match, antibodies in the person's blood can react with the antibodies in the donated blood, and it'll cause the red blood cells to do 2 things: agglutinate and or lyse. Agglutinate means to clump together, whereas lyse means to disintegrate. Lyse being one of those terms we should recognize from a word like hemolyze. We've talked about that in lab before. When this happens, that's called a transfusion reaction, and it can be fatal, and it's very dangerous. So we have to be very careful that we collect our specimens properly. You see these chunks right here, these big clumpy clumps? That's agglutinated red blood cells. And if you imagine that, cannot flow very well. Sorry, hang on. What the hell is happening? Nick, I need you to do more. Faster.
Nothing.
All right, I'm resuming this. Oh, okay, sorry about that. Okay, what was I doing? Oh, agglutinated red blood cells, so you can see that they're all clumped up. Imagine if this was flowing through the body. If they were all clumped up, it's not going to flow very well, and that could be very bad for us. So, we have different blood systems. The ABO blood system is the one that you're probably the most familiar with. And I like this picture because it shows the blood types and how they work. So remember we talked about how your blood type is determined by an antigen on the surface of the red blood cell? So those people with A blood type have A antigen, so represented by these little green triangles right here, okay? People with blood type B are going to have B antigens, which they've represented with little circles right here. Blood type AB has both types of antigens, and blood type O has none. So what you need to know is that people with blood type A, they have this antigen and they make an antibody against anti-Bs. They say we like A's only and we don't like B's, so we make anti-B. So they make little things that go off and kill B antigens. B blood types, they make anti-A antibodies that go off and kill the A antigen. So if they were to come in contact with each other, they would agglutinate and lyse. They would fight each other. That's what those antibodies would do. So down here we have blood type AB, which means these people, they like A's and B's, so they're not going to have any antibodies against either, which means you could give them A and they're not going to fight that off, or you could give them B and they're not going to fight that off. Ideally you give them AB. That's why they're called the universal recipient. As you can see here, universal donor is type O, but they don't have any antigens on their surface, but they do make antibodies against both A's and B's. So they can only receive blood type O, but because they don't have any of these antigens, they can be donated to everybody because there's no antigens on the surface for which there are antibodies to attack. Does that make sense? I hope so. Again, if you need more clarification on this, I can talk about it in class. Feel free to ask me and or go watch some of the other YouTube videos that I've saved on the channel. So, as you can see here, I just talked about that. People with blood type A, they make anti-B. You can see this chart right here. So you guys can look at that on your own. The RH, that's where the positive negative comes from. If it's positive, you have the antigen. If you're negative, you don't have the antigen. That's the D antigen. Most people have received blood that is been ABO, that has been RH typed as well. So for instance, a lot of people always ask me at this point about RhoGAM. It's given to folks that are negative, to women that are negative in case they're carrying a positive baby, because otherwise mom might the first time mom has an RH positive baby and she's RH negative, her body will become sensitized to that and will start creating antibodies, okay, against positive, the RH positive part. And it would attack the fetus's red blood cells, because if mom's negative, it's going to make anti-positive antibodies and it'll start attacking it. So what we do is we give them RhoGAM, and RhoGAM's kind of like a shield. It kind of like hides the fact, right, from from mom's body, it almost like puts this little barrier around them so that they don't see that it's positive and they don't create these antibodies. So this is what happens here when you have an Rh negative and Rh positive baby. This is a nice little representation of that. You don't have to know that for being a phlebotomist, but everyone always asks, so we can talk more about that in class. All right, so blood typing is important because if you're going to get a transfusion, we have to make sure that compatibility testing and cross-matching is done. So compatibility testing and cross-matching is done to determine that the donor unit of blood and the blood of the patient, the recipient, are compatible. So we look at the patient's serum, or sometimes that's if we're drawing it in a red top. But we'll also look at plasma as well, just so you guys know. And they look at that we collect blood from the patient that's going to receive the blood, and they also in the lab will test the unit of blood that they're going to donate to that person to make sure that everything matches. So they look at the ABO types and they look at the Rh positive negative, but they also look at other antibodies that are in existence, although not as popular. But even with all that stuff, every now and then you can still have a reaction. So there's an inherent small risk that comes with getting a transfusion, no matter what. Okay. So now we're moving on away from blood in the body and moving into blood as a specimen. So when we're talking about blood as a specimen, we have two different types of blood specimens. We have plasma specimens and we have serum specimens. So serum is what we're going to talk about first. Blood removed from the body is going to clot or coagulate within 30 to 60 minutes on its own. If it's in a tube that has a clot activator, then it's going to happen even faster. All right. So a clot, what is a clot? A clot is a bunch of red blood cells all held together by this network or net looking thing made of fibrin. And the fluid that's remaining behind is called serum. And we get this fluid out by taking the clotted blood and spinning it down in a centrifuge, right? And I showed you guys that in lab, or I tried to. If I have not already, I can just remind me. So when we spin it down and we get our serum, it's normally a clear yellowish fluid. It looks just like plasma, all right? It is the same composition as plasma, except it doesn't contain any fibrinogen, okay? So since the blood clotted, it used up all the fibrinogen when it made that clot, okay? So a lot of lab tests are done on serum. Plasma is also used just as often. And it looks the same as serum, but basically it's the same thing, except it has its fibrinogen because the blood was collected in a tube that prevents clotting, such as an EDTA or sodium citrate or heparin or a potassium oxalate tube, one that has an anticoagulant in it, which prevents the clotting process from occurring, which means that fibrinogen and the other clotting factors are still present, and that would be called plasma. Okay, so here's a little picture in your book, and it's showing that the red blood cells on the bottom, once this is assuming that you took a purple top and you spun it down, okay? This would be the same in a blue top or a green top or a pink top or any of the anticoagulated blood, okay? So you have your red blood cells on the bottom. You have what's called the buffy coat, which is made up of white blood cells and platelets in the center. You can't often really see that. It's really tiny. So don't expect to see that visually in a specimen, okay? But just so you know, it is there. And then on top, if you had this in, say, a green top tube with gel on the bottom, you would have gel right here above the buffy coat, and then on the top you would have plasma, okay? The plasma is the liquid portion of the blood that's left over, and it contains fibrinogen. What's nice about plasma specimens is that they can be centrifuged right away. You don't have to wait for them to clot, which is why a lot of times you'll see, if something's stat, you'll see it ordered in a green top instead of a red top, because the green top has heparin in it. It doesn't have to clot, so it can go right into the centrifuge and be tested sooner than a red top that has to wait 30 minutes to clot or something like that. Okay, some tests will be tested on whole blood, like hematology tests, so CBCs, things like that. There are some, such as a chemistry test called an A1C, that's done on whole blood, but generally speaking, most tests, or most hematology tests are done on whole blood, but there are some in other departments as well. Whole blood just basically means blood in the same form as it is in the body. You don't have to call it anything else. We can't let it clot. It can't be separated. We just give it right to the hematology department, for instance, and they run the test, okay? Oops, too far. So this is that term, hemostasis, that I mentioned in Chapter 5. There was a word that's very similar. In Chapter 5, we learned the word homeostasis, but in Chapter 6, we're learning the word hemostasis. They mean two different things. They're similar, but different. So hemostasis is basically the body's clotting process, okay? It is the act of the body of stopping the bleeding. It's the body's response to the loss of blood due to an injury, and it's where we convert the liquid, like blood, into a semi-solid gel called a clot. That's the coagulation process. So when a blood vessel is injured, whether it's via venipuncture or you get cut with a knife or whatever the case may be, 4 things occur. First thing that happens is vasoconstriction. The second thing that occurs is the formation of a primary platelet plug, and then we have a hemostatic plug, and then we have fibrinolysis. So very quickly, what does that mean? It means that the first thing that's going to occur is the veins are going to, or the vessel itself is going to constrict and make itself thinner so less blood can flow through that. And then you're going to have platelets all clumped together in form of an initial platelet plug. Okay. Now for venipuncture, this is pretty much as far as it needs to go. For larger issues or injuries, you're going to see that then it goes to the hemostatic plug, and then once that, it's like a big old clot, and you have it scab over, and then when you want that clot to dissolve, we have what's called fibrinolysis occur. So how does all this happen? You have the coagulation factor and pathways. So coagulation factors are types of proteins. They're designated by Roman numerals, like factor 1, factor 3, and they're not in order that they're used. They're named in the order that they were discovered. So once each factor, they activate, right? And then the activation of that factor will activate the next factor, and that's called a cascade. So it's kind of like a waterfall. It goes from one to the next, or like imagine like a series of steps. So you have this cascade then goes into 2 different pathways. You have the extrinsic pathway and the intrinsic pathway. So it's all part of this big clotting process, but it's occurring via outside the cells and inside the cells. If you look at it, like that's what intrinsic and extrinsic means. So you have the tissue factor that happens outside the vascular system. This thromboplastin and factor 3 are located in the cell membranes, and as they're activated, they cause other aspects of the clotting process to occur. Whereas intrinsic is contact activation, and these are factors that are within the bloodstream, and as they come in contact with particular cells in the bloodstream, they activate and then go on to activate other aspects. So they join together in a common end pathway that actually is what forms the fibrin reinforced blood clot. And just so you know, there's 2 different tests that should be familiar, a PT test and a PT test, blood test, that evaluate these 2 pathways. So PT, it actually evaluates the extrinsic pathway, monitors Coumadin therapy. So it lets you know that Coumadin works on the coagulation process somewhere on the extrinsic pathway, whereas PTT, which evaluates heparin, lets you know that heparin works on the intrinsic pathway. So I always thought that was kind of cool. So we talk about thrombin. This is like a major enzyme in the coagulation process, and how it is generated at the injured site from prothrombin. Its primary purpose in life is to convert fibrinogen to fibrin, and fibrin is then used, as you can see, in more of these factors and processes until eventually we get to the clot. You do not have to memorize all of this coagulation, activation, all this stuff. You don't have to know all the different factors. You just need to know like the foundation of how the clotting process works. So here's what you need to know. You need to know what vasoconstriction is. Okay, it's where the narrowing of the blood vessel, as you can see here. Hemostatic process. So you have an injury out here. Let's pretend it's a venipuncture. Okay, the vein is going to constrict so that the blood flow is less coming through this way, so that less blood cells can get out. Then you're going to have a bunch of platelets clump up together and do that and clog that hole. And now for venipuncture, that would be enough. Eventually it realizes that that's clogged, the vasoconstriction goes away, everything's great. If it's a larger injury, you're going to start having a secondary hemostatic plug, which is all this blood clotting, so it's going to start catching blood cells. Fibrin's going to start forming, creating this little net, as you can see here, through all of these activated clotting factors and thrombin and all that stuff here. Okay. Once that clot is no longer needed, then fibrinolysis occurs. Okay, lysis, remember, is the destruction of. So you have this stuff that happens, and it starts breaking down these fibrin things, which lets go of these blood cells, which then removes the blood clot. Pretty cool, right? So this video is also from your book, which we'll go ahead and we'll watch real quick.
Platelet plug formation. Platelet plug formation occurs after an injury to a blood vessel, such as a hole left by a venipuncture needle, exposing substances that cause platelets to stick to the damaged tissue in a process called platelet adhesion. The injury also exposes tissue factor, or TF, which leads to the generation of a small amount of thrombin. The thrombin binds to the surfaces of the platelets, causing them to change shape and stick to one another in a process called platelet aggregation, in which they also attract other platelets to the injury. Clotting factors released from the mass of platelets form more thrombin, which begins the small-scale generation of soluble fibrin threads, as depicted by the yellow strands, entangling the platelets to form a platelet plug. For very minor injuries, like a needle puncture, platelet plug formation and applied pressure are sufficient to seal the site, and coagulation goes no further.
Pretty exciting, right? All right, so like I said, you don't need to memorize all of this stuff, but you are supposed to understand how it works so that it's not sounding like a foreign language to you. So this is a pretty cool picture, I thought, of red blood cells wrapped up with fibrin all over it. Kind of cool. Okay, fibrinolysis, like I said, this is where the fibrin is dissolved. All right, so it shows you this stuff again. The liver plays a big role in the synthesis of these coagulation factors. So factors 5, 8, prothrombin, fibrinogen are all made via the liver, okay? Certain cells called mast cells in the liver make heparin. Heparin occurs naturally in your body, and it works as an anticoagulant in your body, all right? So you can read a little bit about this on your own. All right, and that's it for that. Now we're going to talk about the lymphatic system. The lymphatic system's purpose is to return tissue fluid to the bloodstream and to protect the body by removing microbes, processing lymphocytes, and it also works to deliver fats to the bloodstream. I bet you didn't know that. The fluid inside the lymphatic system is called lymph. It is made up of excess tissue fluid. That's something that most students get wrong when asked on a test. You do need to know what it's made up of. Similar to plasma, but it's a little bit more water. Remember, plasma was 90% water, 10% stuff, whereas lymph fluid is 95% water. And it's made up of these lymphatic vessels, ducts, and nodes. So you can see all of this, okay? Little picture in your book talks about all the different lymph nodes. Usually lymph nodes are depicted in green or yellow, depending on your textbook. You should know that there's nodes and what they do. That the tonsils, thymus, GI tract, and spleen are also part of this system. You have the axillary nodes. We might have already talked about that in class. This is important because this is why we don't draw blood from someone's arm when they've had a mastectomy, because most of the time they've removed these nodes from the armpit, and it can interfere with the lymph drainage and potentially interfere with basically the filtering of bacteria and foreign matter. All right, so excuse me, you guys will talk about this case study in class and the questions and the answers to that, and you can go look at some of the other things on your own time. All right, and that's it.